So earlier, I was talking about lentiviruses, which are retroviruses, and HIV is a lentivirus, and how we're using the ability of lentiviruses or retroviruses to insert DNA into your genome to do the same with the genes that we wish to insert. So this commenter asked, how is this different from CRISPR? Well, the first main difference is that, unlike lentiviruses, this is based on a system that bacteria use. So forgive me, but I'm going to make this simplistic and jump over a bunch of steps. Only about 10 or 20 years ago, we were able to describe a system that bacteria have that is an immune system of sorts. When a virus or a bacteriophage attacks a bacteria, the bacteria has a system that takes small segments of the DNA from that virus and inserts it into a specific region of its genome. This region was given the name of clustered, regularly interspaced, palindromic repeats, or CRISPR. It's the bacteria's blacklist of sorts. So the next time a virus or bacteriophage attacks the bacteria, one that has attacked before, the bacteria has that blacklist. And it can use that blacklist, the RNA from it, with a protein called Cas9 to recognize the virus, and then Cas9 cuts the virus DNA and is very specific. It was only about 5 or 6 years ago that some researchers who won the Nobel Prize, listed them here, described how they can use that system of an RNA of our choice instead of the blacklist from the bacteria and Cas9 to cut specific places in our own genome or the genome of a model organism or the genome of an agricultural plant or animal. After it cuts that location that you have guided it to with the RNA, your DNA repair system will reconnect the cuts. And if you give it a template, like a change in the gene or a whole new gene, etc., it will repair that cut with that template inserted. It's quite a powerful technology and has already been used in research and gene editing for agricultural plants to make them more resistant to disease and to cure human diseases, or at least start to. It doesn't come without problems and issues, both technological and ethical. The technological hurdles are just a matter of time, I think, but the ethical ones are something we really need to consider. For example, do we want to make germline changes, in other words, in the sperm and the egg, that will be passed on from generation to generation? Do we have the right to change the genetic makeup of our descendants, or animals and plants for that matter? Should we make the changes in embryos? Some recent research suggests that there's some big problems with that, but it is powerful and does have a lot of promise.